Oh, how come it's not? There it is. All right. So prokaryotic diversity. Learning objectives differentiate hopefully between archaea, bacteria, eukarya, basic features of bacteria versus archaea, and then how do we classify prokaryotes? Because they are similar, but they can also be different. So let's talk about a brief history of microbiology. That is white. So microbiology intertwines microscopy and disease because prokaryotes are so small you can't see them. And so for most of human history, they were undiscovered. Kind of like mind-blowing when you think about it. This physician, I don't even know how to say that name, Fracastoro, suggested that maybe disease was caused by something you couldn't see, maybe an organism that you couldn't see. Then we had Antony van Leeuwenhoek, who did not discover, or he didn't invent the microscope, he just used the microscope to observe, and he actually did a pretty good job accurately describing microscopic life, particularly in pond water. But there was this idea at the time that life came from non-living things. I know we have discussed spontaneous generation in the past. At least I hope I did. I think it was the PowerPoint with the giraffes and their swinging necks. But just how people thought living things came from non-living things. And so Louis Pasteur set up an experiment to refute the idea of spontaneous generation. You may remember he used like swan neck bottles exposed to air and one that was like vacuum sealed. And the vacuum sealed never developed any microorganisms in the broth. And the one that was opened did develop some type of culture in the broth, suggesting that, hey, there are microorganisms in the air. But Robert Cook, a little bit later, came up with Cook's postulate. And there's 4 statements that microbiologists use, or even like someone that works for the CDC tracking down viruses or bacteria that cause disease. So they literally follow Cook's postulate to this day. So he was studying anthrax at the time and proved that there's a causal relationship between the microorganism as well as the disease. So the 4 statements has something to do with like the microorganism has to be present in every case. So, all these people have similar symptoms. Okay, so then you better like take blood samples or tissue samples, and you have to be able to see or prove that the microorganism was in that individual. Another thing is you have to be able to take that whatever's causing the disease out, and you have to culture it in a petri dish. Okay, because then you could take that petri dish and infect a healthy host. And if they come down with the disease, that's more evidence that, hey, that's the thing that's causing the disease. And then the 4th statement is you have to take that healthy host, they're not healthy anymore, right? And you have to take a tissue sample and affect one more person, or one more organism, all right? And then that proves that's the agent that's causing the disease. Okay, so they still use it today. With the study of microbiology and prokaryotes, we've made some advances in knowledge, particularly our immune system and how it works. And then another thing is that we've also come to the conclusion that archaea and bacteria are probably the oldest, structurally simplest, most abundant life forms. And when I mean most abundant, we haven't even scraped the surface of discovering prokaryotes on planet Earth. It's estimated that we've only characterized maybe 1 to 10% of prokaryotes. And another thing I'd like to talk about is that archaea is relatively kind of like a new domain. It wasn't until so I think archaea and bacteria were grouped together as Monera. So like when I don't know, I've seen old textbooks where it was like eukarya and Monera, but then they had to break Monera into archaea and bacteria just because they discovered that there's some prokaryotes that don't match up with each other. So they had to separate them out. So archaea is, I don't want to say relatively new, it's just that now we have a new division of them, and that prokaryotes are very ubiquitous. They can survive anywhere. So yeah. But they are fundamentally different from eukaryotes. Number 1, they are one-celled, unicellular. However, they do have the ability to live in complex communities of different species, of different prokaryotes in a biofilm. So a biofilm you might find a biofilm in someone's like mouth that maybe they don't have the best hygiene, so they have like a lot of cavities and plaque buildup, or something like that. You see biofilms in like plumbing pipes, like that. I don't know if you've ever had the pleasure of turning on a sink and then you see like this like stringy thing coming out. Well, good. I'm glad you've never had to experience that. And you're like, nope, not drinking that water. But anyways, the biofilms, they're tough to fight. Okay, because they're a community of different species. Biofilms are more resistant to bacterias. They are more resistant to drying out or desiccation, and they can survive through some pretty extreme stressors because they kind of like they're a community and they help each other out. Cell size can vary tremendously, but just know that they are small. I have a picture here of this E. fischelsoni, which is the largest prokaryote that you can see with the naked eye if your eye is very good. So yeah. Okay. Nucleoid, they don't have a nucleus, but they do have a single circular chromosome that kind of is in the cytoplasm of the cell. There are some exceptions, Vibrio cholerae, which I discussed, I think, with viruses, or maybe it was, I think it was viruses. They have 2 circular chromosomes, and then also they can have a nucleoid, but they can also have a plasmid, which is like an accessory DNA molecule that can be transferred between different prokaryotes. Some other characteristics of prokaryotes. They, when they reproduce asexually through binary fission. So there's no sexual reproduction, but they can mix up their genomes through genetic recombination, and that is through horizontal gene transfer, which has been discussed towards the end of our evolution chapter, just like the transfer of genes from like one species to another, or from different prokaryotes to other prokaryotes. Prokaryotes don't have organelles, but they do have some type of compartmentalization produced by their plasma membrane. So there are some bacteria or prokaryotes that can do photosynthesis, and you're like, wait a minute, Mrs. Erickson, you just told me that prokaryotes really don't have organelles. Well, what they do is they use their plasma membrane and they form these invaginations, these little pockets, if you will, and these pockets hold enzymes that can carry out photosynthesis, which is PSN, and cellular respiration, which is CR. So they use their plasma membrane to act like organelles. Prokaryotes tend to have a flagella, and I know earlier this year we talked about the difference between eukaryotic flagella and prokaryotic flagella, but prokaryotic flagella do not have the 9-2 microtubule arrangement. Okay, so they lack that. Their flagella is also rigid and it spins like a propeller, whereas with eukaryotes it undulates like a snake. And then we also have metabolic diversity. There are some photosynthetic bacteria out there, some that produce oxygen, some that do not produce oxygen. And if it doesn't do photosynthesis, then we tend to call it chemolithotrophic.